Hello everyone, I'm Franny Dellacqua, and the topic of this talk will be microplastic ingestion from Atlantic Menhaden collected from differing geographic locations in Charleston Harbor. Brittany Parker and her co-authors published a 2020 study showing that Atlantic Menhaden have very high rates of microplastic consumption, specifically samples in urban estuaries such as Charleston Harbor. This study both incorporates and builds upon the Parker and co-authors' survey of microplastic occurrence in fishes from urban estuaries. The idea that Menhaden are consuming such high concentrations of microplastics in comparison to other fishes is mostly attributed to the fact that Menhaden are plankton filter feeders, and therefore they commonly feed on organic particulates such as marine snow that accumulate very high levels of microplastics. So the referenced microplastic study found previously that fibers were by far the most abundant of the microplastics found, and they also found presence of tire particles in the stomachs of the fish. This research proposes the question, is there geographic variability in the ingestion of microplastics by Atlantic Menhaden in Charleston Harbor? These fish were collected by the South Carolina Department of Natural Resources in 2018 in multiple sampling sites. The three that data from our study come from were Cooper River, Charleston Harbor, and Ashley River. These samples were frozen and thawed 12 to 24 hours before dissection. The digestive tracts of the fish were extracted and placed in mason jars with 1 molar potassium hydroxide and then placed on hot plates until the contents were mostly dissolved. The solution was then poured through a 63-micron sieve, and the contents on the sieve were poured into a vacuum filtration setup in order to capture the microplastics onto the filter for lab analysis. The filters were analyzed with a Leica microscope with varying magnification levels. Microplastics could be identified compositionally with a probe or with the hot needle test, using a soldering iron set to 660 degrees Fahrenheit to test reaction of the plastic. So pictured in this slide is a tire particle and a blue fiber found during the survey. This map shows 3 of the sampling sites of the Menhaden from the data. So the left shows the Ashley River sampling site, and then the Cooper River sampling site, and then the Charleston Harbor sampling site. Parker and her co-authors exhibit in this figure the visibly large difference in microplastic presence in differing species. Atlantic Menhaden showed significantly higher levels of microplastic concentration to gram of fish than the other species, which probes for further analysis of the microplastics within this species specifically. These data were from 3 batches of fish from differing locations, finding that there were far more microplastics per gram of fish found in the Ashley River location, averaging at about 39 microplastics per gram. This could be attributed to the much larger abundance of data from this location in comparison to the other 2, as well as the fact that the fish in this area were generally smaller in size and produced larger ratios of microplastics. Again, the bar graph representing this study showed Ashley River to have the highest overall concentration of microplastics among the 3 sampling sites, and the primary microplastics identified were fibers, consisting of about 87% of all microplastics among the combined data sets, and the average microplastic per gram found among all sites were 8.3 particles per gram of fish, which was actually much less than those found in the Parker study, also likely due to smaller sample sizes from this study and less analysis. This project has not been finished, and there will be a continuation of analysis of the geographic variability in microplastic ingestion for the Atlantic Menhaden species, as this has not been done before. And it's also important to take into consideration the differences of microplastic presence in fishes of different sizes due to differing feeding habits. And it was also interesting in the data to see that there's such a large presence of fibers in all the samples collected in comparison to other microplastics. And it was notable in the 2020 study that there were a large presence of tire particles in these areas as well. I would like to thank my advisors, Dr. Gorka Sancho and Dr. Barbara Beckingham, for their help this semester, as well as the South Carolina Department of Natural Resources for providing samples for the study. And I'm looking forward to finishing off the study for the rest of the year. Thank you for listening, and I hope you all have a good day.